welcome to my channel Anupma Biology Classes. This is the third video in the series of reproduction. Today our topic is fertilization in the flowering plants. Before this, we discussed about the site of sexual reproduction and the formation of male and female gametes in the flowering plants. This video is from chapter 2 of CBSE class 12th. Now, let's start the topic, that is the second portion of the chapter 2. And here the first topic is pollen-pistil interaction. Now the question is, what is the pollen-pistil interaction? So it is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen, and this interaction is mediated by chemical components of the pollen and the pistil. In this diagram, you can see the diagrammatic representation how the pollen-pistil interaction occur. So, after the pollination, pollen adhesion on the stigma surface, then the pollen hydration occur, then pollen germination, then pollen tube entry into the stigma, then pollen tube growth through the style, and then the pollen tube entry into the ovule and embryo sac, and the last double fertilization occur in case of angiosperms. So this is the germination of pollen and the formation of pollen tube, where the pollen grains release its content, and the pollen tube growth through the style of the pistil towards the ovary, and then the entry of male gametes into the ovule and then to synergids. Now here the pollination assists the transfer of required pollen grains from the anthers to the receptive surface of the carpel. Then the pollen grains germination occur on the stigma of the pollen tube through the style up to the embryo sac, and it depends on the interaction between the pollen and pistil. The stigmas of some angiosperm are wet while others are dry. Wet stigmas possess proteins, amino acids and lipids in the form of free-floating secretions, and the dry stigmas possess a hydrated layer of proteins, carbohydrates and lipids. The tapetum of the anther has certain chemicals which carry some specific sporophytic recognition factor, and the chemicals of stigma are also sporophytic origin. So because of this sporophytic origin and the sporophytic recognition factor of stigma, it means pistil, and the pollen, then the pollen-pistil interaction occur. So there is an interaction between the chemicals of the pollen grains and those present on the surface of stigma, and these interactions determine whether a pollen grains will germinate or not. Now the topic is self-incompatibility, which we discussed in the previous video, but here we will discuss this in detail. Now, so what is the self-incompatibility? Actually, the problem is at the time of fertilization, the stigma of a particular plant receives a number of pollen grains, either from the same mating type or from others, and the functional pollen which belongs to right mating type germinate on stigma and develops pollen tube. After that, fertilization occurs, and other pollen grains of different plants are discarded. So, most of the angiospermous plants possess a compatibility to inhibit germination of the pollen grains and further growth of pollen tube, so as to avoid inbreeding and help outbreeding. This chemical control of an individual plant is called self-incompatibility. Now, if a pistil carrying functional female gametophyte gets pollinated with right mating type of the pollen grains but fails to set seeds due to inability of their gametes to fuse with each other, called sexual incompatibility. And you know that the sexual incompatibility are of two types. If sexual incompatibility may be between the individuals of different species, is called interspecific sexual incompatibility, and if between the individuals of the same species, it is called intraspecific sexual incompatibility. Now, the intraspecific incompatibility that occurs between the members of the same species is called self-sterility or self-incompatibility. Now, the incompatibility achieved by first, that is the prevention of pollen germination, then inability of the pollen tube to penetrate into the stigma, then inhibition of pollen tube growth in the style, failure of pollen tube to enter into the ovary or ovule, failure of the fusion of the male and female gametes, and the abortion of embryo following syngamy or development of embryo endosperm incompatibility. So these are some points which shows why incompatibility occur. Now, the self-incompatibility has been divided into two major morphological types, and these are the heteromorphic type and the homomorphic type. So, what are the heteromorphic and what are homomorphic? Heteromorphic is in which the two or more morphologically distinct mating types occur within a species which can be recognized without a breeding test, while a homomorphic is in which the two or more mating types within a species are morphologically indistinguishable and they require proper breeding test. So this is the some differences between the heteromorphic and homomorphic type of the self-incompatibility. Now, based on the factor of pollen grains, self-incompatibility is divided into two types. First one is the sporophytic incompatibility and second is gametophytic incompatibility. In case of the sporophytic incompatibility, if the incompatibility is controlled by the genotype of the sporophytic tissue from which the pollen is derived or the sporophytic tissue of a stigma is called sporophytic incompatibility. While in case of the gametophytic incompatibility is due to genotype of male gametophyte, that is the pollen. So this is a picture which shows the sporophytic self-incompatibility, and here the secretion of stigmatic tissue or the transmitting tissue prevent the germination or growth of incompatible pollen. Now the topic, that is the emasculation and bagging. So, often a plant breeders desire to cross a particular plant with the desired pollen grains. For this, bisexual flower is artificially transformed into female flower by removing its stamens in bud condition. So, what is emasculation? Emasculation is removal of stamens or anthers or killing of pollen grains of a bisexual flower without affecting a female reproductive organ. Here you can see there are two types of emasculations, in which the first you can see by the forceps method and the second by the scissors. And the bagging is after emasculation. The emasculated flower is immediately enclosed in a bag to avoid pollination by any unwanted pollen. So during artificial fertilization, the mature and viable pollen grains are collected from the male parent, then bag is opened and the pollen grains are dusted on the stigma. Then the bag is replaced. So that is the emasculation and bagging. All are occur for the desirable varieties. So this is the emasculation and bagging. Now the main process, that is the fertilization. So what is the fertilization? Fusion of male gamete with the female gametes to form a diploid zygote within the embryo sac is called fertilization. In angiosperms, the male gametes are carried to the egg by a pollen tube called siphonogamy. And this was given by Strasburger in 1884. So, in the process of fertilization, there are many steps in which the first one is growth of the pollen tube. Now, you know that the pollen is two-celled, which for the fertilization reach the receptive stigma of the corbel for the process that is known as pollination. And these pollen grains on the stigma attached and absorbed water and swell up. After this, they start releasing wall-held proteins and other components so that the recognition factors from the pollen and stigma come close to each other. After recognition and acceptance on stigma, the pollen grains germinate to produce pollen tube, which grows into stigma towards the ovarian cavity. In this process, the pollen wants nourishment which was given by the cells of stigma, and then the tube secretes exogenous pectinase and other hydrolytic enzymes to create a passage for its entry. And here, the length of pollen tube varies from few millimeter to 45 centimeter, as in case of maize. So this is a starting point, that is the growth of pollen tube, how the growth occur, and the attachment of stigma to the pollen, that means pollen pistil interaction. After that, now the second step, that is the entry of pollen tube into the ovule. The mature monosporic polygonum type of embryo sac consists of 3 antipodal cells, one secondary nucleus, 2 synergids and an egg cell. The synergids direct the growth of the pollen tube by secreting some chemical substances. And the tip of pollen tube enters into one synergid, then the penetrated synergids start degenerating. After penetration, tip of pollen tube enlarges and ruptures, releasing most of its content, including the 2 male gametes, 7, and the vegetative nucleus into the synergids. So, here in this picture, you can see how the entry of pollen tube into the ovule occur. So, at first, in the first picture, you see here the entry occur through the micropyle, that is from the micropillar end, as in case of the lily. Second, by the chalaja, that is from the chalagal end, as in casuarina. And the third, by the lateral side, that is from the integuments, as in cucurbita. So, if pollen tube enters by the micropyle, it is known as porogamy. If the pollen tube enters by the chalaja, it is known as chalajogamy. And if the pollen tube enters by the integument, it is known as mesogamy. So here, the porogamy is more common and the mesogamy is rare. And after the entry of pollen tube by the micropyle, it pushes the nuclear tissues and finally pierces the egg apparatus end of the embryo sac. So this is the way how the pollen tube enters into the ovule. Now the third step, that is the double fertilization, and this is the most important characteristic feature of the angiosperms. And here both male and female gametes involved in the fertilization. And in this picture you see all the cells of the embryo sac, then release of sperm cells and the double fertilization occur. So the question is, what is the double fertilization? Here, out of both male gametes, you know that there are 2 male gametes in one pollen. So out of both male gametes, one male gamete fuse with the egg and form diploid zygote. And this process is called syngamy or generative fertilization. And this zygote develops into embryo. After then, the other male gamete fuse with the secondary nucleus to form triploid primary endosperm nucleus. Why triploid? Because you know that the secondary nucleus has 2 polar nuclei. So it is 2n condition, and after the addition of extra nucleus by the male gamete, it converted into 3n condition. So the primary endosperm nucleus is called triple fusion vegetative fertilization, and these two act of fertilization called double fertilization. This was first given by Navasin in Fritillaria and Lilium. Now after this, you can see here the complete process of fertilization. So at first you can see here the pollen grain adheres to the stigma, which contains 2 cells, a generative cell and a tube cell. Then the pollen tube cell grows into the style. The generative cells travels inside the pollen tube and it divides to form two sperms. That means two male gametes. Then the pollen tube penetrates an opening in the ovule called a micropyle. So it is known as porogamy. And then one of the sperm fertilizes the egg to form the diploid zygote, and the other sperm fertilizes the two polar nuclei to form the triploid endosperm, which will become a food source for the growing embryo. So this is the complete process of the fertilization. Now, these all are the fertilization events. So, let's come on the post fertilization events or post fertilization changes. So, soon after the act of the double fertilization, the flower begins to lose its color and the petals, stamens, and styles either fall or wither away. But in some cases, calyx persist like in tomato and the brinjal. So, because in the post fertilization changes from the fertilization, the flowers converted into a structure where the petals, stamens, styles are not occur. So, only the development of ovary. So the major events include in case of post fertilization is development of endosperm from the triploid primary endosperm nucleus in the central cell of the embryo sac, then the development of the embryo from the diploid zygote, development of seed from the ovule, and the development of fruit from ovary. So these are the steps in the post fertilization changes, and here the first is that is the development of endosperm. So what is endosperm? Endosperm is a food-laden tissue formed during the development of angiospermous seed, which provides essential nutrients to the growing embryo and also the young seedlings at the time of seed germination. Now, in gymnosperm, female gametophyte is known as endosperm, and in angiosperm, the endosperm develops from the triploid primary endosperm nucleus, which is formed as a result of vegetative fertilization, triple fusion or fusion of a male gamete with the secondary nucleus of the central cell. After this, there are something that is the types of endosperm. And here, the first one that is the nuclear endosperm. In this picture, you can see the development of nuclear endosperm. In example, in case of Eclifa indica, and in this, the primary endosperm nucleus divides by the repeated free mitotic divisions without the formation of walls. It results in the formation of large number of free nuclei in the central cell of the embryo sac. A big central vacuole develops in the embryo sac, pushing all the nuclei to the peripheral cytoplasm. The free nuclei come to lie between the embryo sac cell wall and the vacuolar membrane of the central wall. Then the second phase begins with the cleavage of multinucleate peripheral cytoplasm, resulting in centripetal formation of cell walls. So, most or all of the endosperm is converted into a cellular tissue. It is most common type of endosperm. Now, the second is cellular endosperm. And here in the picture you can see, in case of cellular endosperm, the first nuclear division of the primary endosperm nucleus is followed by the formation of either a longitudinal or transverse cell wall in the central cell. Then the subsequent nuclear divisions and the wall formation result in the formation of a cellular type of endosperm. Here the examples are petunia, dhatura, balsam, etc. So this is the cellular endosperm. Now the last is halobial endosperm. In the picture you can see the development of halobial endosperm. Here the primary endosperm nucleus moves to the chalazial end of the embryo sac. Then the first nuclear division of the primary endosperm nucleus is followed by the formation of the transverse wall, resulting in the formation of a small chalazial cell and a large micropillar cell. Generally, the chalazial cell doesn't divide further and functions as a haustorium. Nucleus of the large micropillar cell divides by repeated free nuclear divisions, and the further development takes place in the same way as the nuclear endosperm. And this halobial endosperm is prevalent in case of the monocotyledons. Now, here the development of embryo, and at first you can see in case of the dicotyledons. So, in case of the dicotyledons, here the zygote enlarges in size, secretes its own cellulose cell wall and divides by a transverse division, resulting in the formation of two unequal cells. Here, the large basal cell is called suspensor cell, which lies towards the micropillar end of the embryo sac, and the smaller terminal cell, which is known as the embryonal cell, which lies toward the chalazial end of the embryo sac. Now, the basal suspensor cell divides by the transverse division, and the terminal embryonal cell divides by longitudinal division to form a tetrad. Out of the two cells formed from the transverse division of the suspensor cell, the lowermost cell towards the micropillar end enlarges in size to form a vesicular basal cell, and the other cells divides by a variable number of transverse division to form a long suspensor consisting of a row of 7 to 8 cells. Now, each of the two terminal cells of the tetrad stage of the proembryo divide longitudinally, forming four-celled quadrant stage. And each of the four cells of the quadrant stage then divides by the transverse division to form 8-cell octant stage. So, in this picture you can see, and then each of the 8 cell of the octant stage then divides by the peritoneal division to form an outer protodermal cell and an inner cell. Consequently, the 8 cell octant now become 16 celled, 8 external protodermal cell and the 8 internal cells. Here, the 8 protodermal cells divides by anticlinal division to form the embryonic surface layer or the epidermis, and the suspensor cell situated at the terminal end of the suspensor is called hypophysis, which you can see in the picture, the position of the hypophysis. Now, the hypophysis divides by transverse and longitudinal division to form 3 tiers of cell, and the cells of the inner tiers towards embryo form the cortical initials of the radicle, and the cells of the middle and outer tier form the initials of the root protoderm and root cap. Thus, all the cells derived from the hypophysis give rise to root apical meristem. At this stage, the developing embryo is globular and shows radial symmetry. It is called proembryo, and the proembryo is transformed into embryo with the development of radicle, plumule and cotyledons. So, the two cotyledons arise from the two ridges of the tissues appeared at the distal region of the proembryo, so that the globular proembryo becomes heart-shaped embryo, and the cotyledons grow rapidly, whereas a few cells between the bases of the two cotyledons differentiate to form a dome-shaped plumule, which is the future shoot apex. So this is the process of the development of embryo in case of the dicotyledons. Now, in case of monocotyledons, in the picture you can see early phase of the embryogenesis up to the formation of globular proembryo are very similar in both dicotyledons and monocotyledons. In monocotyledons, about half of the terminal cell derived to function as shoot apex, whereas the remainder of the cells shows a rapid rate of cell divisions in the formation of single terminal cotyledons. So this is the complete stages in the development of the monocotyledonous embryo. Now, the next topic is the development of seed. So, the double fertilization in angiosperms triggers the transformation of ovule into a seed. Because of this, the following changes occur in the ovule during the development of seed, in which the first one is the zygote develops into an embryo. The triploid primary endosperm nucleus give rise to a nutritive tissue called endosperm. The nucleus is generally used up during the development of the embryo, but in some cases it remains outside the endosperm in the form of a thin layer called perisperm. The outer integument becomes hard and leathery test or outer seed coat, which ensures survival of the seeds. The inner integument, if persists, forms the tegmen. So, the outer integument is known as testa and the inner integument is known as tegmen. The micropyle remains in the form of a fine pore on the surface of the seed. The funicle is transformed into stalk of the seed. The hilum marks the point of attachment to the stalk, and here you can see a complete structure of a dicotyledonous seed which splits open to show the structure, and a longitudinal section of monocot seeds in case of GMAs. So, if the seed have copious amount of endosperm tissue called albuminous, like in case of the wheat, corn, onion, palm, etc., and when the seed in which the endosperm is used up are X-albuminous, like in case of the bean, pea, gram, etc. So these are the structures of the dicot and monocot seed which you can see. Now, the viability of seeds. And in case of the viability, it refers to the ability to retain the power of germination over a period of time. Excessive dry or damp weather and high temperature are known to reduce viability of seeds. Viability of seeds can be tested by two methods. First one is the ability to germinate and the second is ability to respire. Here the question is, what is dormancy? So the dormancy is the condition of seed when it fails to germinate even though the environmental conditions usually considered favorable for the active growth are present. Now after this, the development of fruit. And a true fruit is formed as a result of cell division, expansion, and the differentiation in the ovary. The ovary is transformed into fruit as a result of stimuli received from the pollination as well as from developing seeds. In this picture, you can see the wall or covering of the fruit, that is the epicarp, which is the outer side, then the mesocarp, which is in middle, and the last is endocarp, that is near the seed and that is inside, that means inner wall. After receiving the necessary stimuli, the ovules present inside the ovary are transformed into seeds and the wall of ovary is transformed into pericarp. Now, the fruits divided into two main categories, in which the first one is true fruit, that is eucarp. And you know that the true fruits derived from the ovary of a flower, not associated with any non-carpellary part. And examples like the tomato, grapes, cucumber, pea, mango, brinjal, banana, etc. And the second is false fruit, which is known as pseudocarp. And you know that the false fruit is derived from the ovary along with other accessory floral parts. And the examples are, as in case of the apple and fig, the main edible part of the fruit is the fleshy receptacle. And you know that what is receptacle, which we discussed earlier. Now, what is the role of pollination in fruit development? So, pollination is an important process which is essential for the fertilization, seed development, growth of ovary, and prevention of ovary abscission, that is the drooping of ovary. And now it is well established that the pollen grains released from the specific hormones, which together with the auxins of the carpellary organ stimulate the initial growth of the ovary. And the effect of auxins is including the development of fruit has been observed in tomato, brinjal, tobacco, and several forms of the cucumbers. Now, the role of seeds in the fruit development. Developing seeds play a significant role in the development of normal fruit. It has been observed that a young apple fruit with seeds on all the sides develop normal shape, and if the seeds are present only in one side, only that side of fruit develops well. Now, the development of parthenocarpic fruits. You know that the parthenos means virgin and the carpus means fruit. So, the some plants are able to produce fruits without fertilization, and such fruits are called parthenocarpic fruits. These are seedless or contain empty or non-viable seeds. Like examples, as you can see, the fruits formed as a result of ovary development without pollination, like in case of the citrus, banana and pineapple, they are seedless. Fruits which are formed by the result of stimulation by the pollen, that means pollination, but without fertilization, like in case of the orchids. So there are many examples of the parthenocarpic fruits. Now, the ripening of fruits. After the period of growth due to the cell division and enlargement, the mature fruit undergoes some qualitative changes referred as fruit ripening. And some of the important changes during the fruit ripenings, like the softening of the fruit flesh due to enzymatic degradation of the cell wall, middle lamella, and solubilization of the cell content. Second is like the hydrolytic conversion of the storage material into the soluble sugars, and the transformation of the chloroplast to carotenoid rich chromoplast and the accumulation of anthocyanins give characteristic color to the ripe fruits. You know that the normal fruit are green in color, and after the ripening it converted into the colorful. So, the accumulation of flavoring components contribute to the flavor and aroma of the ripe fruits. So, these are the some important parts of the development of fruits. Now, here the epomixes and the polyembryony. So, at first you see that what is the epomixes? So, the development of reproductive propagules without the meiosis and the syngamy is called epomixes. And the epomixes therefore is the formation of new individuals by the asexual method, which mimic sexual reproduction including seed formation but do not involve fusion of gametes. And there are two kinds of epomixes in the flowering plants. First one is the agamospermy, and in case of agamospermy the seeds are formed by the asexual means, as the embryo develops directly without gamete fusion, that means without meiosis and syngamy. It is of three types, in which the first one is adventive embryony, second is parthenogenesis, and the third one is apospory and apogamy. In the picture you can see the adventive embryony. And what is the adventive embryony? Now, the formation of embryo directly from the diploid sporophytic cells, that is the nucellus or integument, which you can see in the picture, that means nuclear embryos, is called adventive, like in case of the citrus, while the parthenogenesis is the formation of embryo from the unfertilized egg, and the apospory and apogamy is the formation of embryo from any other cells of the embryo sac without fertilization. And the second part of the apomixis is vegetative reproduction, which we discussed earlier. Now, the polyembryony. What is polyembryony? Polyembryony is the phenomena of formation of more than one embryo during the development of seed. And the polyembryony was first discovered by the Leeuwenhoek in citrus. It is of three types, in which the first one is simple polyembryony, where it is due to the fertilization of more than one egg cell in an ovule, like the citrus and pove. Second is cleavage polyembryony, and here it develops due to splitting or cleavage of the proembryo into two or more parts, like the pinus. And in angiosperm, cleavage polyembryony is reported in orchids. And the last is adventive polyembryony, where it involves the formation of additional embryos from different parts of ovule, such as synergids, antipodal cells, nucellus, integuments, etc., like the citrus, opuntia, onion, mangifera, groundnut, etc. So, these are the complete discussion about the process in case of the fertilization in monocots and dicots, structure of seed and fruits, epomixes, polyembryony. And this is the end of the video. Now, the next video is related to the human, that is the human reproductive system. So, if you like this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any queries, any question, any doubt or anything you want to know, ask in the comment section below. Thank you for the watching.